Hello everyone, this is Donna Iula. If you don't know me, I am a synthetic organic chemist and I oversee the forensic chemistry division at Cayman Chemical. I'm so pleased to be part of the conference this year and present a talk titled Synthesis, Development, and Naming of NPS Reference Materials. I realize that title is fairly vague, but what I would like to share today is a brief overview of the steps that we go through from the recognition that there is the need for a new designer drug or NPS standard through all the steps necessary to ship a final certified reference material to a forensic lab. I would also like to discuss some very new and interesting developments on the synthetic cannabinoid landscape. And in reviewing those, you will get to hear about some of the decisions we have to make along the way. For several years now, my presentations have almost exclusively been on the topic of synthetic opioids, so this area is new and exciting for me. Lastly, I will give a quick summary of my perspective on the overall NPS landscape and what new things are going on out there. If you are not familiar with the Cayman Chemical Company, we are located in Ann Arbor, Michigan. We have over 17,000 catalog products. We were founded in 1980, and the work that we do to help make research possible spans everything from GMP manufacturing to biomedical research tools. We have a contract services division, and the forensic chemistry division, which I oversee, is really just a small portion of what Cayman Chemical does. All the work that we do to stay on top of the constantly evolving NPS scene and its various structural classes, and to actually execute the completion of those final reference materials are all carried out by the forensic chemistry division. The crew pictured here is composed of incredibly talented and dedicated scientists, mostly synthetic organic and analytical chemists. And many of them, like myself, have backgrounds as medicinal chemists in the pharmaceutical industry, and some have forensic chemistry and toxicology experience. It is truly a pleasure to get to work with this team every day. Once we have identified that there is a new standard needed, the first step is the design of a synthetic route. Often, newly trafficked designer drugs take their inspiration from existing journal and patent literature, so we always start from there and try to find a known existing route. If we cannot find precedent, then we perform a retrosynthetic analysis and develop a plan to build the molecule up step by step from commercially available raw materials. The execution of the route in the lab is most often the most time-consuming and variable part of the journey. Sometimes what we design on paper doesn't translate into reality in the lab. Many times we have to go back, redesign a route, or perform months of R&D to understand what's going on. A synthesis can take anywhere from a few weeks to several months in some cases, for maybe even a year or more for more complicated metabolites. And along the way, the synthetic chemists characterize their intermediates and their final products to ensure success. The material, once synthesized, then moves out of the synthesis lab into the appropriate quality control group to reassess the identity and assess the purity. For this example, shown on the right, which is an ISO certified ampuled certified reference material, method validation is performed. That lab, the ISO quality lab, will independently characterize the material. Our ISO production lab will then prepare an ampuled solution to a specific concentration. That solution is then QC'd. Stability studies are initiated. There is also, on occasion, regulatory paperwork to fill out. There's material safety data sheets and certificates of analysis to be compiled. And before the launch of the product on our website, we also try to make sure that we have all the necessary pertinent information together to present on the product landing page. This includes relevant references, synonyms, INCHI codes, smile strings, etc. As I said earlier, this can be a long process, and there are often many bumps in the road. It can be very rewarding, but at times it can also be very frustrating. Here are some pictures of the staff in action in Ann Arbor, making and characterizing the materials. My plea to the audience is to reach out and let us know what you need as soon as feasible so that we can get that long process started. You may not realize how important your input is in how we prioritize our work, but it really is important. I mentioned that I wanted to focus on what is new on the synthetic cannabinoid landscape for 2021. As you can see from the DEA's mid-year emerging threat report for this year, all of the synthetic cannabinoids detected, not surprisingly, have been of the carboxyindole and carboxyindazole structural class. This report highlights that ADB-fubiata, which is the 4-carbon tail version, was detected by them for the first time in this reporting period, and the analog MDMB-4EN Pinaca was the most prevalent. The CFSRE and NPS discovery team's third quarter trend report mirrors that same trend. The indole-indazole carboxymids make up the majority of the synthetic cannabinoids that are being detected in the United States based on their work. And they also make up the majority of what they are recommending for testing in the United States. As you know, the synthetic cannabinoid NPS are also prevalent outside the US, especially in Europe. The UNODC and the EMCDDA have been keeping their eye on the increased adulteration of vape and e-liquids and other products consumed by cannabis users. These products are often adulterated with synthetic cannabinoid receptor agonists, or SCRAs, such as the MDMB-4EN Pinaca analog that I just mentioned. This poses a big health threat because often the users of these products do not know what is in them. In May of this year, the UNODC announced, as I issue this announcement as I show here, to raise awareness of this issue on the international level, and also announce that MDMB-4EN Pinaca was placed under international control in April. In May of this year, the Office of China National Narcotics Commission announced that it will place several structural classes of synthetic cannabinoids under generic control. This went into effect in July, and I immediately started to wonder if this act would have a similar effect to what happened after China moved to control the generic fentanyl scaffold in 2019. That is, would we start to see the emergence of new scaffolds to circumvent the regulation? If you haven't read through the details yet and had time to wrap your head around what this actually covers, the announcement includes 7 Marcuse structures. Using R1 through R5, X, Y, and Z to note points of variation, they specified which structures are covered. In reading the details and studying these structures, it reveals what specific analogs are covered under this, as well as what specific analogs are not covered. For example, let's take X. By saying X represents N or C, this means that if you have a synthetic cannabinoid with a nitrogen or a carbon in any of these structures at the specific position noted by X, it is covered under this control. If X is something other than a nitrogen or carbon, it is not controlled. I'm not going to review all of these, but I thought it might be helpful to translate each of these 7 generic structures into at least one recognizable and known synthetic cannabinoid that would be covered under this. So if you take the top row, the leftmost Marcuse structure in the red rectangle covers the majority of the first generation syncans, like JWH-O18 to the newer indole, aza-indole, and indazoles that we see now. The second structure over covers the 2' alkylated indazoles. The third generic structure in the red rectangle covers the gamma-carboline 1 ones, like the cumulpegoclone that I have drawn in the upper right-hand side of the slide. The fourth structure covers the pyrazole core synthetic cannabinoids with an example just to the right. So now if you move to the bottom row, highlighted in that red rectangle, the generic control extends to a variety of carbazoles, such as EG2201 and this cyclohexyl methyl compound. The central Marcuse structure covers the benzimidazoles, such as fubamina. And lastly, the control extends to the cyclohexyl phenols, such as the CP compounds, with CP47, 497 illustrated in the lower right. So that Chinese ban went into effect in July, and what have we observed since then? In August of 2021, we started to get inquiries from forensic labs from several different countries with data for a substance that had never previously been encountered. These images are screenshots of the EI mass specs that we were sent, and our help to determine what that structure might be was requested. The data all had the same pattern. The 109 fragment stands out as a fluorobenzyl piece, which is observed in a lot of synthetic cannabinoids, and details around those seizures led us to assume that this was a previously unidentified synthetic cannabinoid. The mass 395 was exactly 14 higher than a known synthetic cannabinoid, so we presumed it to be this structure on the right, with the 14 corresponding to the addition of a methylene group to the known syncan ADB-fubiata. And prior to this point, we had never seen a synthetic cannabinoid of this nature. So this new compound resembles ADB-fubiata, which I have shown here on the left. ADB-fubiata is a 3-carboxy-indole synthetic cannabinoid that was first reported in 2016 in a forensic toxicology article. As you can see, the carboxamide carbonyl is directly bound to the indole core in the ADB-fubiata structure on the left, whereas in this new compound on the right, there is a CH2 or a methylene group in between. This is the only difference between these two compounds, and this results in the increase in mass by 14. So if you compare this new structure with the Marcuse structures I showed earlier that are covered in the Chinese structural class band, you will note that this is not covered. The amide carbonyl of the indoles covered in that legislation are directly attached to the ring system. So ADB-fubiata is covered, while this new modification, while slight, is excluded from this generic band. Is this newly discovered substance a result of the ban, or is it purely coincidental? It is hard to say. So what do we call this new substance? Rob Shulkin, our resident synthetic cannabinoid expert at Cayman, Alex Kratulski from NPS Discovery, and I started collaborating on what we thought the name should be for this new substance. ADB-fubiata was named in that 2016 paper by pulling out letters from the IUPAC name. For the new substance, we wanted to extend the use of that semi-systematic naming system for synthetic cannabinoids to this substance as well. Obviously, modifying the structure results in a new IUPAC name, so the lettering and number sequence will be different. But we wanted to ensure that the name that we chose for this new substance was close enough to ADB-fubiata to make it readily recognizable as a close analog of it. So after a series of brainstorming sessions over Teams, Rob, Alex, and I settled on ADB-fubiata. Apparently, there was an independent discovery and report of this new substance on a Russian site called VIII in August. They took a similar approach in terms of sticking to that semi-systematic syncan naming system as we did, but they went with FUB-ACADB So we are now unfortunately left with two names out there for this new substance. At the end of October, this drug testing and analysis paper appeared, which describes the identification and structural elucidation of 3 synthetic cannabinoids post-SYNCAN structural class ban. This novel structure was mentioned in this publication. They, however, refer to it as AD-18. So this methylene analog was profiled along with 2 other substances that I will talk about later. And this publication was the work of the Drug Intelligence and Forensic Chemistry Lab of the Ministry of Public Security in Beijing, China. So where did the name AD-18 come from? This item is sold on the gray market or the recreational user sites as AD-18, and from what I can gather, this name is what it is being marketed under on those direct-to-user websites. Here are screenshots of 2 sites that we discovered in the past few weeks. The site on the right advertises this as a new designer drug cannabinoid, quote unquote, developed as a modern replacement for ADB-fubiata. So where did we get the name ADB-fubiata for this new methylene analog? Naming is a big issue in the forensic community, so this decision required a lot of deliberation. The semi-systematic SYNCAN naming system began around 2012. At least I believe this was around the time of the first use of that system. This forensic toxicology paper from 2012 out of the Uchiyama lab at the National Institute of Health Sciences in Tokyo, Japan, mentions the identification and structural elucidation of 2 cannabimimetic compounds, A-pica and A-pinaca, that would represent the beginnings of this new generation of indole, indazole 3-carboxamides as designer drugs. The shorthand names A-pica and A-pinaca came about by pulling selected letters highlighted in red out of the formal chemical names. The ICA portion, the ICA of the shorthand name, represents the indole carboxamide core linker, and the INACA, the INACA, represents the indazole carboxamide. Another 2012 paper from the same Japanese lab mentions the identification of yet another 2 examples of this new generation of synthetic cannabinoids that also uses that naming convention. This paper describes the first identification of AB-pinaca and AB-fubiata. The paper pointed out that the AB-fubiata had appeared in a 2012 Pfizer patent describing the legitimate pharmaceutical research, but yet the recreational drug version was not named as a Pfizer compound using their company internal numbering system in the article. And personally, I think it is wise to adopt a new name in forensic circles that distances the materials from the legitimate pharmaceutical development work. And here again, you see that those names were derived by picking out specific letters from that IUPAC or formal chemical name. So let's get back to ADB-fubiata. How did we arrive at that? Let's look back at the 2 most closely related compounds, ADB-fubica and its indazole version, ADB-fubiata. Those names are generated by pulling out the corresponding head, core, tail letters from the formal chemical name that described the structures, as was described by that Japanese lab back in 2012. ADB is the head, the letters FUB describe the tail, and the I indicates the indole, and the CA for the carboxamide linker. The indazole version is similar, except more letters are pulled out to differentiate them. Hence, the INA are pulled out. When naming SYNCANs, we think of them as having a head, core, tail, and linkers. As I mentioned earlier, we wanted a name that closely resembled the related ADB-fubiata. Just as it is easily understood when hearing the ICAs and the INACAs that you are moving from an indole to an indazole, we felt that having the name end in "ata" would convey that same sense of this is similar but slightly different. The ATA letters were pulled from the formal name were pulled from the formal acetamido portion of the new linker. When thinking about this name, we also planned for the eventuality that, well, could we encounter the indazole version? And so when thinking about that, we planned for that eventuality, and we envisioned that we would recommend ADB-fubiata for that analog. I do not believe that the cannabinoid activity of this substance has been reported yet, but if it is active, we do need to be prepared for the eventuality of seeing methylene versions of other synthetic cannabinoids. The use of the quasi-systematic SYNCAN naming system is not easy, and so I have provided here a link to our wall poster. You can request from our website a large size poster for your office or your lab, and it has all the recommended letter codes for all the various head, cores, and tails. If you want to learn more about NPS naming in general, here's a plug for the naming webinar that Alex Kratulski and I did back in July for the Forensic Technology Center of Excellence at RTI. For more information on this new substance, the drug testing and analysis article I mentioned earlier has a full characterization and supplemental data for the compound that they had found in seized materials, and that data includes EI mass spec, FTIR, ESI QTOF mass spec, and a host of NMR studies. The reference standard is available on the Cayman Chemical website as product number 35059. This slide shows the DMSO, the deuterated DMSO proton NMR for the ADB-fubiata that we synthesized in our lab. The characteristic signal that differentiates this compound from the ADB-fubica is this AB quartet that you see at around 3.6 ppm. This is that isolated methylene group. It appears as an AB quartet because those protons are diastereotopic due to the fact that there is a chiral center on the molecule. Earlier on in my presentation, I mentioned that the start of our journey from the realization that there is the need for a forensic community, need in the forensic community for an authentic reference standard, begins with designing a synthetic route. It turns out when we scanned the literature to see if the synthesis of this compound had been previously described, we could not find one. So this compound appears to be an entirely newly designed substance for the purpose of recreational use. We found that this indole 3 acetic acid starting material was commercially available, and that provides two possible routes to access the desired standard. Route A, shown on top, inserts the head group first and the tail in the second step, while Route B attaches the tail group on first, then functionalizes the carboxylic acid. Both 2-amino-3,3-dimethylbutanamide and 4-fluorobenzyl bromide are readily available. For the production of our standard, we evaluated both routes and optimized the chemistry to for the product over the course of several weeks. For the forensic toxicologist listening in, you might be wondering how this might metabolize. Well, if this behaves similar to other terminal amide syncans with the FUB tail, you might suspect that this will hydrolyze to the corresponding carboxylic acid. That's just pure conjecture on my part, but based on what we've seen with other synthetic cannabinoids, we anticipate that that would probably be the primary metabolite. We already have the synthesis in progress, and we hope to have that standard available sometime in December. I mentioned earlier an October 2021 drug testing and analysis paper, and that they had described 3 new types of synthetic cannabinoid analogs that were not covered under the generic definitions. The other 2 compounds in that paper, which they refer to as MDA19 derivatives, we were also aware of prior to that publication as well. You might have noticed this announcement by the CFSRE and NPS Discovery and Cayman chemical shown on the right here, where we described the identification of and naming of 3 new synthetic cannabinoids. We coined this new generation an overall class name of oxazids. And that announcement came out in August, so it's very recent. And I would like to talk about those and how we arrived at those names. Starting early this summer, we started to receive spectra from forensic labs around the world for these new substances, and we started to see posts on user forums for this new series that we would later refer to as the oxazids. The spectra and chat forums pointed in the direction of these new substances being related to something called MDA-19. We would eventually figure out what these new substances were and share that information with those who reached out to us. And along the way, of course, we collaborated with Alex over at NPS Discovery. So let's start with what is MDA-19. MDA-19, drawn here on the right, is an N-alkyl ecitine acyl hydrazone derivative designed by the MD Anderson Cancer Center in Houston, Texas, and described in a 2008 Journal of Medicinal Chemistry paper. It was designed to treat the CB2 receptor and was a lead compound in their effort to find effective treatments for neuropathic pain. Even though it was evaluated and optimized for CB2, it still has affinity for CB1, which is what gives the SynCan its psychoactive properties. The synthesis of this compound and many analogs were described in a series of scientific publications starting around 2018. In looking at the structure, you can see the resemblance to other synthetic cannabinoids, but with some key differences. This class of cannabinoids, as far as I can tell, is not covered in the Chinese structural class ban. The MD Anderson Center, the MD Anderson Cancer Center, in addition to publishing several scientific journals, have a patent covering these hydrazone compounds which modulate cannabinoid receptors. In this work, they made dozens, if not hundreds of analogs, of which MDA-19 is one of them. The synthesis of MDA-19 is relatively straightforward from readily available materials, so I can see why this is also an easy target for clandestine labs. As described in their publications, these cannabinoids are made in 2 steps if the starting appropriately substituted icetine compound is commercially available. The tail piece is put on using the right alkyl halide, and then the final step is condensation with a hydrazide derivative to make the final hydrazone. For MDA-19 specifically, and what we did at Cayman to make the standard was to treat icetine with hexyl bromide, isolate that intermediate, fully characterize it, and then once we had that intermediate with the tail piece on, we then treated that with benzoyl hydrazine to give MDA-19. The first 3 oxazids appearing in forensic casework this summer have only differed by the tail. The first 3 we have seen have been the hexyl, pentyl, and 5-fluoropentyl tails, which we named, and going from left to right, BZO-hexoxazid, BZO-poxazid, and 5-fluoro-BZO-poxazid earlier this summer. So how and why did Cayman and NPS Discovery come up with the oxazid naming? Well, first we needed to, because not all of the MDA compounds were given numbers, such as 19, and some of them aren't even covered in their work, which is why we saw them refer to these compounds as pentyl MDA-19 and 5-fluoropentyl MDA-19 in that DTA paper. So we asked ourselves, does it make sense to keep using the naming system for legitimate neuropathic pain candidates and referring to every new analog as a variant of this one named MDA-19? Does it even make sense to use MDA in the forensic community due to the fact that MDA is better known as methylene dioxyamphetamine? We asked ourselves all these questions, And again, I collaborated with our synthetic cannabinoid experts at Cayman Chemical and Alex Krotulski from NPS Discovery. And this is the system that we developed. And I can explain it using the 5-fluoro compound as an example. Similar to how we divide up the other syncans using the IUPAC Uchiyama style naming system, we wrote out the formal name, divided the compound up into head, linker, core, and tail pieces, and decided which letters we'd pull out to designate that piece. In this example, the red letters stand for the tail, the blue, the core, the yellow, the linker, and green, the head. If you refer back to that August announcement, you can see how this system works. MDA-19 can then be renamed BZO-hexoxazid with the letters HEX for the fact that there is a hexyl chain. The 5-carbon chain analog in the center was named BZO-poxazid, where the P stands for the pentyl. At the time of that August announcement, there were only 3 oxazids that we were aware of in casework. And between then and now, here in early to mid-November, we have presumptively identified 2 more compounds, the cyclohexyl methyl analog and the 4-EN pentyl variant in that short period of time. Here's a look at our EI mass spec from our GC mass spec for the pentyl variant BZO-poxazid. As you can see, the exact mass, 335, is present. And what we have seen with these oxazids is that the next predominant fragment results from the cleavage of the acyl hydrazone CN bond, which is right here. Cleavage at this spot leaves the fragment 230, which corresponds to this portion of the molecule. We see that similar cleavage of this acyl hydrazone CN bond with the 5-fluoro-BZO-poxazid analog, and cleavage at that position affords the dominant fragment that you see at 248. And also, the exact mass is observed at 353. For more analytical data, you can refer to that drug testing and analysis paper that was just published a few weeks ago, which actually contains an 11-page supplemental data packet with images for all the spectra. Analytical reports, as I have provided links here, can also be found on the NPS Discovery website. The analytical reference standards are now all available for purchase on the Cayman Chemical website. We have been trying really hard to get these standards out there as quickly as possible so that you can make the positive confirmation for cases that you encounter. In addition to those synthetic cannabinoid classes that I've mentioned, there have been other new cannabimimetics on the market. Those have more of the slight modifications to the previously known scaffolds. For example, in 2021, we saw the introduction of a new hexyl tail, which is the hexanaca, which I have at the top of the list here. We've also seen a phenethyl tail, which we call the fetanaca, to highlight just a few of the new syncans for 2021. I'd like to talk a little bit about the use of binaca. If you recall earlier, I mentioned this forensic toxicology paper from 2016 because it was where ADB-fubiata was discovered. Well, if you recall from that slide, the title of that article also has the compound ADB-binaca in the name. And this was the publication that first described the structural elucidation of this compound. And if you see from their journal article, they used binaca to refer to the benzyl group. So the B stood for benzyl. So this is why when we issued our reference standard for ADB-binaca shortly after that paper appeared, we used the term ADB-binaca. So as I mentioned, ADB-fubiata has been out for several years as the benzyl version. So when we started to see this butyl chain variant on the illicit market, we named that butyl chain version ADB-fubiata. And we did that because we had to differentiate the binaca from the butyl chain. So we pulled out BUT from the name butyl to differentiate the ADB binaca from the ADB butanaca. And I know that this has caused a bit of a confusion because some people have referred to the butyl analog as ADB binaca, probably not realizing that there was already a compound out there called ADB binaca, and that had the benzyl group on it. So we realized that this has caused a lot of confusion. So what we have done was made the decision that what we would do was any new compound that we see out there that we have to name that has a benzyl off of that indazole or indole, we would call that a benzinaca. The area of stimulants and hallucinogens covers a wide variety of structural classes, from cathinones to several aryl cyclohexylamines to tryptamines and LSD analogs, and we have definitely seen a lot of activity in the space. There's no time to go over all of these, but I wanted to just get some of these newer analogs that are on the NPS market all on one slide so that you can refer back to them later. 2021 brought some new benzodiazepines and a few new synthetic opioids as well. We haven't seen as much diversity in this space as we had seen in previous years, but I do have the new ones that we have at least heard about or have introduced in our catalog in 2021 for your future reference. I have on this slide a few synthetic byproducts that are and contaminants that are found in fentanyl synthesis. There's a compound on this slide, dipionone, which I have in the upper right column. When I mentioned earlier that sometimes, there's a lot of bumps in the road in the synthesis portion of our work, this is definitely a compound that has caused a lot of blood, sweat, and tears for us. But we're actively working on it and hope to have made some improvements to the synthesis soon. So overall, the assessment is that the drug traffickers and clan labs continue to get more creative and more bold. The open web is full of sites selling drugs for recreational use and precursor chemicals. Having something new to sell or use seems to create a lot of buzz on drug user forums. This site was interesting because it is trying to be clever to disguise the name isodonidazine by calling it isotone space tazine powder, presumably to avoid being detected if law enforcement is doing keyword searches on the open web. This particular open web illicit site sells lots of controlled substances, non-controlled recreational drug substances, as well as a lot of drug precursors. These sites, such as this one, seem to come and go all the time. The screenshot that I have here for this particular product happens to be the starting material to make a number of nitazene synthetic opioids. This vendor site, Trust Chemical Shop, is openly selling something I thought we had heard the last of back in 2017. Remember something called Gray Death? Someone on my team sent me this link recently, and I was shocked at how brazen these traffickers are. Gray Death was found back in 2017 to contain a mixture of U47700, carfentanil, other fentanyls, heroin, among other things. It was causing a lot of overdose deaths, and yet they are openly offering to sell this to anyone. It's unbelievable that they openly admit in these screenshots that I have here that this is deadly. They admit that they don't even know what's actually in the mix. Yet they mention how to consume it, and they throw in that quote unquote, this delivery process is confidential. I know a lot of effort has gone into trying to shut these sites down, but it just really must be hard to keep up with these sites. To conclude here, as I mentioned earlier, to curb the manufacturing, trafficking, and abuse of synthetic cannabinoids, China implemented a class-wide regulation on synthetic cannabinoids in the summer. This has led to the identification of a new methylene spacer analog, ADB-fubiata, as well as a new series called the Oxides. As we continue to see during our surface and dark web monitoring, the clandestine labs and the sellers of illicit and gray market drugs and precursors are getting increasingly creative and at times quite bold. Thanks to everyone for attending this workshop. Thank you to the organizers of the workshop and the conference for including me in this amazing program. I need to acknowledge the Forensic Chemistry Division for all their hard work and for being such a pleasure to work with. I need to give a big shout out to our synthetic cannabinoid expert, Rob Shelkin, for helping me get better at SynCan naming and helping us overall stay on top of all the latest developments in this space. My email address is on this slide, diula@caymanchem.com. Don't hesitate to reach out. Shoot me a message if you encounter something new or you just have a question about naming, you want to talk about naming or synthesis, or if I can help you get that standard that you need. Thank you so much, and I look forward to our Q&A.